Okay, so the tertiary structure has crisscrosses. These are the mini services or pockets for these are active sites. Okay, and where the substrate fits, and once the substrate fits, like the enzyme changes its when the substrate fits, like the enzyme alter its shape, that fitting more tightly the substrate. Now the now when there is close proximity of the substrate, that breaks the chemical bonds of the substrate and the enzyme product is formed. When the enzyme product is formed, the enzyme releases the product of reactions and enzyme is free now for the another cycle. So this is enzyme plus substrate, then enzyme substrate, then complex form, then enzyme product form, then enzyme plus product. Now all these are like transition state, all these are intermediate structures, and all these structures are unstable. Okay, now stability is something related to energy status. You see, the substrate when the substrate has higher energy than the product, then the reaction is exothermic. When it means like energy will be released, and if the product at the higher side and substrate at the lower side, it means endothermic, that is energy is given to the system. Now in y-axis there is potential energy, in x-axis progression of energy. It is activation energy with enzyme, activation energy without enzyme when the barrier is large. You see, now what enzyme does, enzymes eventually bring down this energy barrier, which is activation energy, which is required for transformation of substrate to product. So you see, making the transition of substrate to product more is, so this is it, and there is transition test in between. You see, remember this diagram now. um, how do enzymes bring about such high rates of chemical conservation? As I told you, now glucose converts into 2 pyruvic acid, like 10 10 intermediates are formed, 10 different enzymes are included. Okay, so this is it now. And there is like carbonic, another example is carbon dioxide plus water, the CO2 and H2O, they form the carbonic acid, that is H2CO3, and this carbonic anhydrase. Okay, this is the chemical reactions. Now another point is that, as I told you, like some hydrothermal, like the enzymes are more stable at temperature like 80 degrees celsius to 90 degrees celsius. These are hot winds and sulfur springs. They are very high temperatures, and enzymes are very active at optimum temperatures and active at optimum pH, right? And these are thermostability thermophilic organisms. These enzymes are